Good afternoon, everyone. Thank you very much to the organizers for this opportunity to speak. So I'm Jishnu. I'm an assistant professor at the Center for Systems Immunology at the University of Pittsburgh. I'll be talking to you about a very interesting project where we used a network-based approach to identify expression modules underlying rejection in pediatric liver transplantation. Uh, Overall, my lab uses multi-omic integration to uncover correlates and mechanisms of immune regulation using both predictive and interpretable machine learning. And we also think a lot about not just machine learning on the data, but taking into account the structure and topology of the underlying biological networks, both the protein-protein interactome network as well as the gene regulatory network. And a lot of my past work has done predictive modeling to generate immune correlates in the context of HIV, malaria, tuberculosis. But what we really need is to move beyond predictive to interpretable machine learning. And that's a theme I'll be talking to you about today. So 3 papers from my lab published over the last 3 months really try to move from predictive to interpretable machine learning in different contexts. Obviously, I don't have the time to present all 3. I'm going to be talking to you about this story that we published in Cell Reports Medicine in April, where we use this network-based approach to identify expression modules underlying rejection in pediatric liver transplantation. So this was work done with Rakesh Sindhi, a pediatric liver transplant surgeon at the UPMC Children's Hospital of Pittsburgh. So why do kids need liver transplants? They need it for a wide variety of reasons, from cirrhosis to tumors to metabolic disorders to acute liver failure to biliotresia to cholestasis, and a whole host of reasons. This is already very unfortunate. However, the most unfortunate part is that even after the transplant event, rejection occurred in at least 46% of recipients once. And this is acute cellular rejection. So a lot of people come and tell me, why do you need all of these markers? Can't you just do HLA cross matches? Well, HLA cross matches work in, for example, kidney transplant in adults, which is primarily antibody mediated. But this is acute cellular rejection in a pediatric population where there is no HLA cross matching done. There are no biomarkers, blood-based, that we have. The key point here is we are trying to use blood-based biomarkers. So this is a schematic where we have these kids and we are looking at transcriptomic signatures from PBMCs in the peripheral compartment. Of course, if we sampled at the liver or near the site of transplantation, if we had tissue-based signatures, the signal would be through the roof. But this is in the peripheral compartment and they are on immunosuppressive drugs. So the signatures are strongly dampened. So what happens if we take a standard gene-centric based approach? Well, if we take a standard gene-centric based approach, we did it. And we did this at 3 different time points. I'm just showing you the pre-transplant time point, which is perhaps the clinically most actionable time points. We identified around 70 differentially expressed genes. We used a linear model, same old stuff, correcting for the standard covariates. We found these 70 genes. But when we tried to do cross-platform validation of the top 70 DEGs that we had at stringent statistical significance thresholds, only around 10 of them replicated on a Fluidigm platform. Things look even worse when we try to do computational validation. So if we do nested k-fold cross-validation with permutation testing on all the DEGs or the DEGs that passed replication in the Fluidigm platform, things are really, really bad. So it fails stringent technical replication. So we said, well, there is clearly something going on here. The effect sizes are really weak. This is in the peripheral compartment. We need an approach that takes into account all of this. So rather than using a gene-centric approach, we propose to use a network or a module-centric approach. And the idea is very simple. We combine the expression data with modules derived from topological clustering of the underlying protein-protein interaction network. And we use modules rather than genes as decision boundaries. So we have the reference human protein-protein interaction network. This is from all experimental. There are no predicted interactions here. So these are from high throughput experiments, both mass spec and E2 hybrid. We topologically cluster the network using this published algorithm, cluster 1. And we then overlay expression data onto these and create these modules. And then we use L1 regularization to down select specific modules. This achieves 3 things. First, modules are functional units. So we are getting additive effect. So the dampening that we had, we are trying to overcome it by taking into account several genes in the same functional process. This is a standard idea in systems biology, but this is really implementing it in a pediatric liver transplant context. The second is we have two levels of dimensionality reduction. The first level of dimensionality reduction is biologically inspired, where we are going from genes to modules based on the underlying structure of the protein-protein interaction network. And really, these genes are not independent. The protein products encoded by them are part of this underlying protein-protein interaction network. And then we have L1 regularization, which is statistical dimensionality reduction, and we are down selecting these modules. So this is all great. What happens in practice when we implement it? When we implemented it at the pre-transplant time point, we actually now start seeing signals. So this is, again, the performance of the model across 100 replicates of nested k-fold cross-validation. The AUCs are not great, but at least going from no signal, we are at the 0.65 to 0.7 range. One question that often comes up is, what if you use pathways? If you use pathways, we don't really get any signal because pathways are likely overbroad. So these modules are much more specific. And with regularization, we are picking very, very specific modules that can serve as decision boundaries. So what are these decision boundaries? They take into account the entire spectrum of things, from type 1 interferon signaling to complement to cell proliferation, DNA replication. And at least this is the first step. I'm not claiming that this is the most interpretable, but rather than having a random list of genes, some of these processes are already targets of existing immunosuppressive drugs. So we can do something about this. So diving more into this idea, this is what the heat map of the different modules that we picked up look like. The rejectors, if we do a partial least squares discriminant analysis, the rejectors have a relatively tight profile in terms of how they co-vary among themselves. The non-rejectors have a very, very different profile. But what is actually more interesting is that these network signatures are druggable. So they are targets of a lot of drugs. But of course, going from bench to bedside, if you really do drug discovery or drug repurposing, that's 10 years. What is most interesting is some of these modules are targets of existing immunosuppressive drugs. So I will give you 3 examples. Number one, and these are 3 different patient profiles that we saw who underwent rejection. So, and this is, again, at the pre-transplant time point before the event occurred. So if you have only higher module-specific scores for complement type 1 interferon or mTOR signaling, you can simply add a third agent, often done clinically like rapamycin. Profile 2 is you can have higher module-specific scores for additional things. For example, in addition to these complement and like inflammatory processes, type 1 interferon signaling, you could also have dysregulated cell cycle DNA replication. You could add CellCept, which is, again, already used in the clinic. And the third is if you have across the board higher scores, that means that not any specific targeted therapy, but you would need a polyclonal agent to deplete all lymphocytes. So this is, again, preliminary, but if we see replication of this signature, this is a viable strategy that we can take. And the other part of this mechanistic thing is, of course, this is at the pre-transplant time point, but at the post-transplant time point, when we look at it, the signatures change. It goes from essentially entirely innate to adaptive, and we see things like AID-induced DNA damage repair, which is related to somatic hypermutation and affinity maturation, so suggesting this is not really, that's the dogma in the field, that it's pure acute cellular rejection, but there are probably components of antibodies involved. So it's really a change from innate to innate and adaptive. So in summary, we've uncovered interpretable module-centric expression signatures from peripheral blood. That's the key thing. This is actionable, directly actionable, because you can collect these samples that underlie rejection in pediatric liver transplantation. We have demonstrated that network module signatures are more predictive than individual gene signatures. And this I showed you at the pre-transplant as well as the post-early. We've also done it post-late. And we identify which modules can be targeted by current anti-rejection drugs, enabling personalized adjustment of drug regimens for dominant targetable pathways. So this is my lab. I started my lab in January 2020. It's grown rapidly. All of this work was driven by a fantastic postdoc, Saeed Rahman, who've highlighted in red. And a big thank you to our funding agencies. We've been very fortunate to have a lot of support from different funding agencies. We have funded projects across infectious disease, HIV, influenza, autoimmune disease, scleroderma. So we are really looking to hire at all levels, especially postdocs. And a big thank you to our collaborators, Rakesh Sindhi, pediatric liver transplant surgeon, as well as Mailarappa Ningappa, who along with Saeed Rahman, a postdoc in my lab, co-led this project. And thank you very much to all of you for your attention and for the invitation to present. Happy to take any questions.
You probably found, I don't know, dozens, hundreds of modules from using your clustering algorithm. So, did you select which module would go into your classifier?
Yeah, so good question. To give you concrete numbers, when we did the topological clustering, the total number of modules was around 500 to 600. But using L1 regularization for the pre-transplant time point, we narrowed it down to around 13 to 15 modules shown here. So these were all the modules. And at the post-early time point, we are actually down to only 3 modules. At the post-late time point, we are down to around 5 modules. So we are really using stringent regularization to come down to between 5 to 15 modules, not using all the modules.
How do you learn the structure of the modules?
That's a great question. So in this specific, and this is something we've given a lot of thought to, and I kind of glossed over it. In the specific case, we've done topological clustering using a published algorithm in Nature Methods called Cluster 1, which maximizes the intra-module connectivity and minimizes the cross-module, its overlapping modules. So this is simply topological. The clustering is being done invariant of the expression data. We are now working on approaches to do the joint clustering, which would perhaps be better, because we are saying these are the functional subunits, and then we are treating the modules and the number of modules as hyperparameters we optimize, but we could do it together. The second non-trivial part to it is, I glossed over it, but how do you sum up things in the same module? Because they could be in the same direction, they could be in opposite directions. So we did a bunch of different feature engineering steps, and it turns out that no matter what we do, additive, non-additive, trying to take into account basic epistatic effects, the models don't exceed an AUC of 0.65 to 0.7, which probably tells us something about the actual signal going in. We even tried nonlinear approaches, but I think we need better signal. I mean, bulk RNA-seq is kind of the tip of the iceberg. If we have additional data modalities, I think we would improve much better.
Yeah, thank you. I enjoyed your talk, and just following up on that, I was curious whether you've had a chance to look at whether there were particular cell types that were enriched, and or whether it's M1 or M2. So that sort of.
That is a great question. That is a great question. So this is from bulk. We haven't done the deconvolution, but we do think there is, and this is in a separate project, we do think there is, although this is acute cellular rejection and the dogma would say it's all T cell driven, we do think there is a role for B cells. I mean, we see AID-induced DNA damage. We also think there is modulated antigen presentation and processing, which is from some other data which I didn't show. So dendritic cells could also be playing a role. Thank you.